are nice and green. So he came to us to talk to us about how to get the proper biology in there and would that indeed fix the problem with the diseases and the weeds and those sorts of things. So onto the surface of the golf course, of the greens, over the surfaces he put one application of compost, a good fungal dominated compost, as fungal as we could, because what he was missing were the fungi in the soil. There were no fungi to be found, there were no protozoa, there were no good guy nematodes, and therefore that's what needed to be fixed. He had bacteria out of the zoo. Not a problem with bacteria. But he's missing everything else in that food web. So an application, 1 ton per acre. That is the barest dusting on the surface of your soil. And so on everything in the golf course, so all, what is it, on the golf course is like, the greens are only 18 acres or something like that. Ridiculously small. And then the fairways too as well, a ton of compost. And then 3 applications of compost tea went out. So once a month for the next 3 months. The first application of compost started to bring the food web up to the balance that we required, but it didn't get it there. The next application of compost tea, with all that biology and a little bit of food along with it, moved things in the right direction a little bit more. The second application of compost tea moved things very close to just about right. Third application of compost tea, bingo, got it. Golf course was beautiful. He's still there. He's kept his job. And so here we are, about 6 months after he started, looking into the soil on the greens. And you're looking at all that great fungal hypha. So good structure, movement of water through there. The water doesn't puddle anymore. People on his golf courses can start playing after a 1-inch downpour of rain. They can be back on the greens 20 minutes later without any damage. So very happy about this, and he goes around England talking about this.
Is this just a straight photo, or is this microscopic?
This is a straight photo. This is just with his little digital Nikon camera. So you can see these with your eyeballs. Same thing with that picture. You can see these with the naked eye.
What was the time for the application?
1 month. Between the compost went on, I think the compost went on in February, and then the next application, the first application of compost tea a month later in March, the next application of compost tea in April, the third application of compost tea in May, and no diseases, no insect pests, no weeds, reduced his costs massively. On this golf course, he estimated that they probably saved $1 million in the first year. So, was the Greens Committee happy with that? Which tells you how much money were they spending on pesticides and inorganic fertilizer and mowing and water if he saved $1 million in the first year. It is frightening. I've gone to like National Golf Course on Long Island and asked them, how many pesticides do they use? And they always go, oh, we don't hardly use any pesticides compared to the guys across the street. Well, then you're not going to mind if you give me the list of pesticides that you put on, are you? Oh boy, that was like trying to pull teeth. Finally, they did. Those guys are putting some kind of toxic chemical out at least once a week. Once a week. No wonder. What is it? 3 out of every 4 women on Long Island have or will have breast cancer during their lifetime. 3 out of every 4. It used to be 1 out of 10. It's 3 out of every 4 now. Whoa. If you live on Long Island and you're female, you will have breast cancer.
Wow.
Whoa. On Hawaii. Probably nowhere close, right? But think of the toxic chemicals that they're exposed to on a daily basis. On their golf courses, they're putting something toxic out once a week. And that was National Golf Course. And they said they used way less pesticides and inorganic fertilizers than the guy across the street. Could we get that information from the guy across the street? Not a chance. So, golf courses, yeah. That's pesticide city.
How could they even make such an assertion?
They obviously knew something about what they were doing across the street. So it's a little frightening to think about that kind of information. Now, data, but we actually have evidence, well, we couldn't get it. So when we start talking about question,
You know, when we were looking for the name for this workshop, and we made it Common Ground, and we said that when the committee, you know, there were a few of us, they said, you know, the basic of agriculture is the ground, you know, and so if you're a renter, you're whatever it is, it's the ground, and that's why we brought Dr. Elaine out here. But you know, I look at what any agriculture industry can use the practices that she's talking about. The only issue that I see is a real larger issue is guys who's exporting plants. You know, to me, I think, you know, because the way, I don't know why, you know, hell that we can't use our own soils in Hawaii to export. Any plants that's going to go out, it got to be soilless, right? And that's one of the issues that we have with a lot of the nursery guys. So, but other than that, I think for crops, for trees, for pasture, for what have you, you know, when we looked at common ground, I mean, this is one of, I think, among all the workshops we've been doing, this is one of the most, to me, intriguing and beneficial for everybody. So I just wanted to say that, I don't know how we're going to mitigate that, you know, so that, you know, exporters, like the biggest nursery over here, export nursery on this island is right here in Kohala, you know. And then I thought, hey, you want to come to me first? Oh, what for? Because I cannot ship anyway. If I, you know, if I gotta go through to that, you know. But I don't know. You know, maybe we can. But then you question, where does the cinder come from? Right?
yeah.
The cinder comes from Pahoa. You know, it's not soil. You know, but where does it come from? You know? So anyway, I just wanted to bring that up because I think sometimes we only think about this as only for crops or only for trees, but it's common ground for all.
When we were talking about hydroponics, we're starting to do tea pines, and the plants are growing way better than the chemical system. So we work with most of the greenhouse growers in the state of California, and they're all switching over to using biology because it's way less expensive, and it doesn't destroy water quality if you don't have all of the problem chemicals leaving once or twice a day, because you start getting those really high level. We can go into hydroponics units and we can have some fun with that really fast. So maybe a different time, but it's. You know, another thing to think about is, do we want to be shipping our soil anywhere? No, so how do you raise your plants for sale on the mainland? We really want to grow those plants in compost. Let's put it into pure organic matter. And we can control the disease-causing organisms. We do not have any of the disease-causing organisms in a properly made compost. And you have several choices of the way you make your compost to be 100% guaranteed that you have no pathogens and no pests, no root-feeding nematodes in those composts. They're lighter than soil, so they're not as expensive to ship. We don't have the problems with compost as we do with soil, and yet we'll raise just as good, if not better, plants. Because the important part is the nutrient cycle. Protection is that list of 5 things that I went through literally first thing this morning. So we have to get to a point where we're really understanding how to do this. Who are the organisms, and then how do you get those organisms back into whatever you're trying to grow your plants in? We shouldn't be sending soil Hawaiian soil should stay in Hawaii. You know, who's the goddess that doesn't want any of the rocks to leave?
Heather.
Yeah. So does she want any of her soil to leave? So why use soil? Not necessary. So we've been through this concept of bacteria make the glue that hold the small pieces together. Bacteria glue the sand, the silt, the clay, the organic matter. They glue themselves onto those surfaces and start making them micro aggregates. And then fungi are going to be the mortar that holds each one of those little micro aggregate bricks together. And they build larger structures so that we're allowing oxygen and water. So kind of think about soil structures being like a brick house. The bacteria make the bricks. The fungi are the mortar that holds the walls and the floors and the ceilings, and they decide where the windows are going to be and where the doors are going to be and where the drains in the floor are going to be and where are the skylights and where are the holes, the structure that allow oxygen and water to move together, move through that soil. Let's oxygen move through that soil as deep as it possibly can. If we compact soil, these are the organisms that start the job of uncompacting your soil, putting structure back into it so your water can go down as deep as it possibly can. Oxygen can go down as deep as it possibly can. How far down into the Earth's surface can oxygen move? Is soil actually anaerobic some distance down? No. We can have anaerobic pockets in the soil where we've had so much organic matter that the bacteria growing on that, the fungi, drive that anaerobic, and that's what we will eventually someday call an oil well. But those are unique little pockets, and some really weird things happen in order to have those conditions occur. When we go looking, and we really understand how far down into the soil surface does oxygen move, oxygen moves as far as you want to measure it. Some of the work we've been doing with oil wells, with oil companies, we have oxygen moving down as far as 21 miles into the surface of the Earth. Do we find active, living, growing bacteria and fungi all the way down there? Absolutely. Aerobic bacteria and fungi. Now you get down at 21 miles and your rocks tend to get a little molten. They get to be a little fluid because they're under all of that pressure. Do we find active, living, growing bacteria, fungi, protozoa, and nematodes at that depth in the soil surface? And they're down there, they're happy, they're growing. There's some work that's coming out of University of Aberdeen and the Macaulay Institute in Scotland, showing that there are active, living, growing bacteria in molten lava. Woo. Just because we've never looked doesn't mean they're not there. So we've only begun to understand all of this biology and what it's doing. They're there, we need to understand. And then how do we manage, or how do we use them? How can we go work hand in hand with them? Fungi are the organisms that are condensing the simple plant materials to ever more complex materials. And so when we're looking at a picture like this, and we can see the fulvics and we can see the humics, those humics are condensed plant materials. Once upon a time, this was a plant. Can you tell the plant from which this organic matter came from? It's been chewed up many times by bacteria and then by fungi, making it ever more complex, ever more gnarly. And it is the savings account of mineral nutrients. So another source of minerals. If you've got organic matter in your soil, forget the fertilizers, as long as you have the biology. So now we've talked bacteria and fungi. Let's go on to the things that eat bacteria and fungi. So predators. So the things that eat fungi and bacteria are the protozoa, eat bacteria. And so 3 groups, we've talked about ciliates already, so you know what they look like. This is a flagellate. So flagellum. Look at the number of bacteria in that soil. See all the aggregates? How would you like to count those at this dilution? So to count the bacteria, we're going to dilute more. But we're going to be looking for flagellates at this dilution. So here's a flagellate. Typically he has one flagellum coming out this side of the body, and he has another flagellum coming out of that side of the body. And they can be kind of hard to see because they're using those flagella to motor around in the soil. So this particular one, he's got a flagellum that's kind of rotating and pushing him through the soil. And the flagellum at the other end, he's grabbing onto stuff and pulling himself along. Grabbing and pulling and motoring. So this guy's moving pretty fast through the soil. Well, he may have to be moving real fast to chase down those bacteria. Because a lot of bacteria are motile too. Zoom, zoom. So bacteria tend to have real sharp angles that they turn. Flagellates don't. They're fairly bigger, fairly much bigger. They are bigger than bacteria. So think about the individual bacteria here. Now there's a couple of bacteria. There's an aggregate. There's another aggregate. A single bacterium, a single bacterium. There's the flagellate. There's the flagella. There's a bacterium that the flagellate has eaten. So this is flagellate motoring around eating bacteria. Well, as these flagellates eat bacteria, they are going to be releasing plant-available nutrients, meaty as well. So they release nutrients in a plant-available form. How does that happen exactly? Well, we will talk about that probably sometime tomorrow morning. We'll get down to that gory level of information. Presumably you do want to know it. So it takes a few more facts to understand exactly why when bacteria and fungi are eaten by the predators, they're going to be releasing plant-available nutrients. So just hold on, we'll get there. But here we're trying to look at these different kinds of materials in soils. Just going off on a little bit of a side note, a lot of people will ask about mycorrhizal fungi. And there are mycorrhizal fungi that are inside root systems. They're not actually inside the root. They're inside the root system. And these mycorrhizal fungi colonize your roots. So we're looking at active exchange sites of what the fungal hyphae going off into the soil, collecting nutrients, pulling it straight out of your rocks, your sands, your silts, your clays, your organic matter, pulling those nutrients into the fungal hyphae and translocating them back to the plant. These arbuscules, there you go, arbuscules, are the exchange sites of these nutrients coming from the soil. The fungus says, hey, plant, you want these great mineral nutrients? And the plant says, yeah, absolutely, I want those. Here's some sugar in exchange for those mineral nutrients. So when we take your roots, wash them off, put them under an epifluorescent microscope, we can immediately see if you've got active exchange sites, these arbuscules, exchanging nutrients with your plants. So, looking at these kinds of interactions between organisms in the soil and the benefit to the plant. Now, let's go to another predator. So this is Elaine. Isn't she cute? Okay, you have to be a nematologist, so you can imagine my husband is a nematologist. He is the nematologist for the Pacific Northwest. And he has the charge to deal with all of the nematode problems in Oregon, Washington, Montana, Idaho, Northern California. So he has a little bit of work to do. So here I am, a microbial ecologist. I work with bacteria and fungi, protozoa. And here's my husband, nematologist. And so he works with identifying all of these different kinds of nematodes. And how do you tell and what do they do? So you can imagine our dinner time conversations at my house. It's probably fairly scary, which explains probably why both of my children are in the world of entertainment. My daughter works for ESPN and my son is an opera singer. We scared her today. With all the who's who and all of the problems going on. So now when we're looking at the nematode, nematodes are pretty huge, because look at the individual bacteria, those are aggregates. But here's this nematode. At 400x, these are large organisms that are pretty much the size of your field of view. But how do you tell if it's a good guy or if it's a bad guy? You gotta look at its mouth. So here's a nice big mouth, but it doesn't have any spear. There is no tooth in here. They've got a couple of little hairs, so whiskers on their chinny chin chin. And the only thing that can be is a bacterial feeder. So when this bacterial feeder eats bacteria, it's going to release nutrients in a plant-available form. Some of its favorite foods are the disease-causing bacteria. And they tend to home right in on disease-causing bacteria and wipe them out. They will also tend to push root-feeding nematodes away from the root system. Because this nematode knows that most of the bacterial foods in soil come from the plants. And therefore, it's concentrating most of its effort and time right around that root system. And if a root-feeding nematode comes and eats the root, where's the food for this bacterial feeder? It's bright enough to know how long has these interactions been going on in soil. For the last, about 2.5 billion years. So there's been some time for some selection to happen. These guys know that root feeders are bad news. And so if we have decent levels of these bacterial-feeding nematodes, they will prevent the root-feeding nematodes from finding the root system. So plants are putting out these exudates. And the bacteria and fungi grow on those exudates and form a castle wall around the root systems. Of course, the place where you have most of the bacteria and fungi in soil growing is right around that root system. Here's the highest concentration. If you were a bacterial-feeding nematode, a fungal-feeding nematode, where are you going to find the smorgasbord? Around the root system. So most of the predators in the soil are going to be concentrated in that root system. So bacteria and fungi growing on the cakes and cookies that your plant is producing attracts the protozoa and nematodes, eat those bacteria and fungi, make those nutrients available right back to your plant. Who's in control of that system? Plant. By the exudates it's producing. Can your plant change the type of exudates that it's making? Today I need more calcium, so I'm going to put out the cakes and cookies that grow those calcium solubilizing bacteria, fungi. So I'm going to grow a herd of these bacteria and fungi that are taking the calcium right out of my sand, right out of the silt, right out of the clay, right out of the rocks and pebbles. And that's going to attract the protozoa and the nematodes, the microarthropods, the earthworms. They eat those bacteria and fungi and deliver to my root system. Thank you. Have the calcium that I need.
Is that in their excrement, basically?
It's, yeah, from the predators, it's in predator poop, if you will. And we'll go through a little bit more detail on exactly why and how that happens when we get into a little bit. So when we look at concentration of these nutrients in the soil around those root systems, we're producing excess of what your plant requires, typically. And so if your plant's only taking up 1/10 of those released nutrients, what happens to those soluble nutrients that your plant doesn't take up? Do they leach out of the root system? Now remember, your plant's feeding cakes and cookies to the bacteria and fungi. They've got to have these soluble nutrients themselves. So who's waiting in the wings? If the plant doesn't take up that soluble nutrient, now who's going to take it up? Right back into the bacteria and fungi. So we don't have any leaching from any soil that has a good set of microorganisms in it. if we've got the proper balance, you don't lose any of your nutrients. So if we can have this good biology in our soil, we don't get leaching into our groundwater, into our surface waters. We need to hold on to those nutrients. We need to keep them in the soil. We do not want them leaching into our drinking water, because then it's going to cost money for you and I to clean that up. Fungal feeding nematodes. We'll take a look at more of these when we go into the microscope day. But here's a fungal hypha. Really wide diameter. We're looking at about 12, 13 micrometers in diameter. Here's our root feeding nematode. He has a spear. And he uses that spear to punch a hole through the cell wall of that fungal hypha. And then you can see what he does. He sucks the internal contents of that fungus out. So, you know, nice little fungal hypha. Here comes the root feeding nematode. Sitting right there, takes that spear, that stylet, and punches a hole in that fungal hypha and sucks out the internal contents. See, isn't soil a great place? Ooh. What would you like to be? Fungal feeding nematode or a fungus? So, you know, these guys are mobile, the fungus is not, so he's kind of like, ugh. So, fungal feeding nematodes have spears. And so if we're looking for those fungal feeding nematodes, we want to find simple spears. So we differentiate between something with just a simple mouth versus something that's got a spear in it.
And no whiskers on these guys.
Now this is a predatory nematode. It has a big mouth, bigger than our bacterial here. Predatory nematode. Doesn't sound like that's got to be bad. It's predatory. But what kind of things does it eat? It eats other nematodes. So are there, what do we call those, cannibals in the soil? Nematode that eats other nematodes. But these predatory nematodes, great big mouths, and they always have teeth. They're pretty easy to see because they're just such honking big nematodes anyway. So here's another nematode. It kind of went up to the front end of this other nematode and went and started pulling it in. It kind of averts that tooth and grabs on, pulls them in. Pulls them in. So just like a little kid eating a piece of spaghetti. Gone. Find another one. When we're dealing with most predatory nematodes, their absolute favorite food are the root feeders. They do home in on root feeding nematodes. So there has to be something that root feeding nematodes are putting out that attracts these predatory nematodes to come and chow down on. Root feeding nematodes, what are they just full of? All kinds of good nutrients that eat your roots. So they are focused on, they will home in on your root feeding nematodes and help you wipe out that problem.
Are root feeding nematodes and the fungal feeders the same?
Root feeding nematodes and now root feeding nematodes only eat roots. Fungal feeding nematodes eat fungi. Bacterial feeding nematodes, what do they eat? Bacteria. So they kind of, the name is a giveaway. Predatory nematodes eat other nematodes. Here's a root feeding nematode. Root feeding nematodes have spears, but look at the structure of this spear. See the big knobs? Because when you're a root feeding nematode, you've got to have a spear that can punch its way through that honking big cell wall of that plant. So you better have something for the insertions of your muscles. So you can pull this spear back and punch through that cell wall of that plant cell. Real thick, real big. Cell walls of fungi aren't that thick. They're not that hard to get through. So fungal feeding nematodes don't need big, nasty spears. Root feeding nematodes do. So we can almost invariably see that we're dealing with a root feeder when you're looking for that spear and the knobs. So in our little microscopes, we will typically be able to differentiate between root feeders and the beneficial nematodes. We will spend more time on all of those morphologies on Friday. So hopefully now when I start talking about root feeding nematodes, you're going to get not spear in your mind. When I talk about predatory nematodes, big mouth with a tube. Fungal feeding nematodes, just a simple spear. Bacterial feeding nematodes, just a simple mouth. When we deal with protozoa. Now, flagellates, amoebae, ciliates, you can differentiate. We've got some movies that I want to show you so that some of the way these organisms move, you wander around in this one. Here we go. I want to show amoebae because we haven't looked at them yet. So there's an amoeba. See how it oozes? And this guy is real jerky the way it's moving. So there's the body of the amoeba. See all the bacteria inside there? And it puts out this pseudopod, a fake foot. And so it's going to move in that direction, but the pseudopod followed by all the rest of the material. And look at this. It's bringing along a bacterial aggregate just in case it runs out of food. Bring along the lunch pack with it. So there it's happily oozing its way. You can quite often see it kind of engulfs the bacterium, and next thing you know, that bacterium is inside of its body.
Is that in real time?
This is pretty much in real time. Yep. Where are you? Go. Where is it? We gotta look around and find it. There he is. So, you know, he's oozing along. He didn't like that, so he's going this way instead. So we'll watch him for just a few minutes so you get a feeling for this movement. When your soils are really warm, these amoebae can be moving this fast. When you're dealing with cooler conditions, when the temperatures aren't as warm, they tend to slow down. So they can ooze really slowly, so you're going to have to be looking at anything that kind of looks like it might be amoeba. See if it moves. If it's moving really fast, it's easy to find it, but a lot of times they're just not taking off that fast. You know, and just watch for how it engulfs bacteria. Now it's inside, and it's going to take that amoeba a little while to digest that bacterium. All these different kinds of materials. Now, as this amoeba keeps moving along and keeps doing its thing, you're going to start to see vacuoles that develop. So see these little holes, these little kind of clear spots in here, and they'll get bigger and bigger, and then they'll just disappear, because that's amoeba poop. That's amoeba manure being released out into the medium, and that's where your soluble nitrogen, soluble sulfur, all your plant available forms of nutrients are coming from. So watch the vacuoles develop, and then they'll just disappear. So getting a feeling for that movement. So is he going to engulf those bacteria, or is he going to slide right on by? All of these protozoa have preferences. There's some bacteria that they love, and there's some bacteria they don't want anything to do with. Spit them right back out. So as he's moving across that slide, you can see that he leaves stuff behind, and other stuff that he picks right up and goes and starts consuming. So, yeah, he can go right by stuff that he doesn't like. And so each species of amoeba has different preferences. So they're not typically going to wipe out all your bacteria. So one single amoeba like this in a drop of water, you can see where he's not going to be wiping out most of your bacteria. But what if we had 1,000 of these amoeba in here? Could we do a pretty good job? Though that many amoeba do a pretty good job of wiping out all your bacteria? Oh yeah. We don't want too many, we don't want too few. Goldilocks principle. Everything in the soil is balanced. We want enough, but not too many. Goldilocks walked into the Three Bears house. Sat down at the table on the first bowl of porridge. Ooh, too cold. Left it alone. Next bowl of porridge. Ah, too hot. Left it alone. Third bowl of porridge, just right, so she heats it up. Same thing here. We want just the right balance. Enough to release nutrients, but we don't want too many. So there goes our amoeba, wandering on. Now let's go towards the end of the picture. How do we go? Oh, what's this? Too small. See, notice the golden green color in there? That's an algal cell. It's a single algal cell. Algae come in a lot of different sizes and a lot of different shapes. So you do kind of want to learn what they look like. A nematode would have a esophagus, it would have intestines, it would have a anus, it would have definitely mouth parts, a tail, and that thing is just bilaterally symmetrical. It's an algal cell. There's our amoeba happening, moving along.
It's carrying around the cell.
Yeah, it's there's some bacterium in there that has glued that algal cell onto the rest of that organic matter. So the amoeba's just dragging along with her. Just trailing along behind. So we're looking at a spore of something here, probably a fungal spore of some kind. We have lots of smaller fungal spores, so we can be aware of spores of fungi. We do not count spores because they're not active, they're not performing a function. Just look, that amoeba is engulfing something. Right there. Do I want to eat it or not? I'm not sure. I don't know. I want to go this way. No, we're going this way. No? Come on, you're going this way. The conversations that must go on. So, this guy really wanted to go that. And there's a vacuole. Now this is one single amoeba. Amoeba will binary fizz. They undergo binary fusion. So if that's binary fission, then the verb would be fizzing, isn't it? Microbiologists aren't sure about that. So let's keep going. What else are we going to find? Oh, right here at the end. Let me go. What the heck's going on here? So here's our amoeba zooming on. Now look at how all this stuff in this water is all of a sudden just zooming, just moving really fast. And look at this. What happened? It's the edge of the drop of water. It's drying up. So when you're looking at a sample, and all of a sudden all of your critters, all of the things in your sample started, oh wow, they're just really fast that way. They're all moving together. They are not developing some new kind of motility. Your drop is drying out. Take your slide apart, put another drop on there, and you're good to go. Okay, so what other fun stuff can I show you? Let's look at just some cute protozoa wandering around. So now see these guys all zooming around? Whoa, is this an active sample? When see this little guy as it's going along, see how it bumbles? It has a sideways motion to it. So see him bumbling along? And there's a definite hitch in the skid along as it's moving. Those are probably flagellates. Even though we maybe can't focus, we can't use our iris diaphragm to see those two little flagella on it, that bumbling motion tells you that those are flagellates. How can you tell for sure? Okay, now take a look at this guy. See all the little hairs there? Cilia. So look at all the hairs. So this is higher magnification. We're up at about 800 total mag. But we're looking at those hairs, the cilia on his body. So lots of cilia over here, more over there. Have a grand old time with bacteria. So here we are back to that guy. That's a flagellate going by as he's bumbling along. Now, see this critter here? He's kind of like an accordion. He stretches out and he's eating stuff, and then he'll pull back in, and then he'll go over here and eat some more. Insect larvae. Insect larvae. Mama fly was attracted to this area because of the anaerobic conditions. And so she laid eggs in here. The egg hatches and starts to grow. So where we're seeing insect larvae, we know that it's not so wonderful. Can you see that amoeba move? It's way slower, isn't it? Can you find the part that's moving? Over here, that oozed a little bit. See how slow they can be sometimes? Sometimes you just have to be real patient to look for that kind of motion. Okay, some more flowers to look at. You don't need to listen to much. So there's a leaf, and it's time-stop photography, time-lapse photography. So there's the fungal hypha coming up to the edge of that leaf material. The fungus is what starts decomposing plant material, wide C/N ratio. As the fungus chews on it, it releases materials that are now bacterial foods. So you can see how that bacterial biomass developed over time. But when we're decomposing leaf material, when we're decomposing woody materials, it's always going to be fungi first coming to decompose this material. And so over time, this is about 3 days, you can see those fungal hyphae growing and just completely engulfing. Now, these are fuzzy-wuzzy fungi. I'm not real happy with but it is wide diameter, so it's probably not a real bad guy, but it is clear, so it's yeah. And then along come all the bacteria. Bacteria always eat things from edges. Bacteria can't attack things in the middle. So the more surface area we can develop, the more rapid the decomposition by bacteria is going to go on. As we shred these materials, we're going to get more bacterial growth, maybe a little bit less fungi. So here's some controlling factors that we can deal with when we're looking at decomposition. Let's keep going. Okay. Let's look at, in a laboratory condition, we're looking at a root hairs. So don't confuse these for fungi. They're usually quite a bit wider in diameter than fungi. There is a blue color, blue stain in this liquid. And so as the plant is growing, it's pulling that blue stain in, and you can see where it concentrates. Material that a plant takes up by simple diffusion. That material is pulled in through the root hairs into the root system, and those materials accumulate in the root itself. These plants, they are growing in gelatin material, and you can see that these are not real heavy plants. So I'll go back. Let's go back just a little bit. Okay, so there was a couple other things I wanted to point out. So our root hairs, here's a bacterium. So you can see this is about 800 total magnification. There's our 2 bacteria, this is about 400 mag. Now the root hair is much wider than any other fungi. Not real healthy plants. Did you see how anemic and unhappy and so restricted oxygen going through that medium. Root is putting out exudates, throwing billions of bacteria. Look at the castle wall being formed by that root system. Now you are seeing sloughing of the root cells, And you can see how the root cells are accumulating. Bacteria don't decompose sloughed roots or root cells. So you see they got pop off? So as those are cells, those are individual root cells. And nothing in this sample, nothing in this material to decompose those sloughed root cells. And then as the oxygen is used up by that massive amount of bacterial growth around that root system, look at what comes along to eat the bacteria. And what were those organisms we were looking at there? Want to give it a guess? So blue stain being pulled in, putting that blue stain down in the root system. The plant's going to have to modify this and turn it into something else in order to keep pulling in any of those nutrients. So we have individual bacteria on here, bacterium, high mag. We go to a little bit lower mag, right there. So uptake of nutrients by those roots. Not real heavy plants. Not adequate oxygen movement in here. So right at first, the exudate's coming out of that root system, growing a castle wall of bacteria. But the sloughed root cells are accumulating. We don't have the fungi in here to decompose those sloughed root cells. And to get rid of those, we've got to get some fungi growing. Now, as you move back along that root, we're going to look at a few more cells sloughing away. Did you see it pop out? So it's a way of the root easing its way through whatever medium it's growing in. Gelatin like this isn't offering much resistance, but think about soil.
The bacteria in the cells.
Yeah, inside the cells. Now, so what are these? Can you see cilia all over the outside of their body? And they come in and out of focus. All of these are ciliates. Or what? Ciliates. So what does that tell you about the oxygen conditions in this medium around that root? So why is the plant real unhappy and not growing well? So that's the problem with so many of our experiments that have been done in laboratory cultures. There's absolutely no recognition that these plants are sick, they're unhealthy, they're growing in an anaerobic situation. So there's no normal nutrient cycling going here. If we're doing experiments on how plants take up materials when they're sick, unhealthy, not happy plants, is perhaps some of the data we've been getting from these kinds of experiments. Just a little question. So how do roots grow through soil? And so these pictures are from a Japanese lab where they have done all this time-lapse photography. So well-structured soil, looking at the aggregates in here. And that root is moving through that soil and encountering lots of air spaces. No problem with oxygen or water moving through here. Now hold on, watch, pay attention, look. Look where the root hairs actually started growing. It's a long distance past that tip of the root. So here's that root pushing its way, and it is sloughing root cells to help it move through there. And it's the fungi holding those aggregates together that are going to utilize those sloughed root materials. Oh yeah, we got a castle wall of bacteria, we got a castle wall of fungi around that root. So thinking about how roots actually interact with organisms in the soil, it's really important to recognize
Do the clumps also provide space for oxygen to get down there? The clump creates airspace for the oxygen, and that's why it's healthy.
a lot easier. Think if we didn't have the bacteria and fungi making those clumps, all of that material would be equally dispersed. And so water moving through all those little tiny spaces, it would take forever. You would have no drainage through your soil. How easy would it to, big rainfall happens, it's going to back right up, right at the surface of your soil. Oxygen does not move through water very well at all. They go anaerobic very rapidly. But if we have those clumps, bacteria and fungi pull them together. Now there's all these big air spaces, channels, living rooms, dining rooms, swimming pools, and for all of that oxygen and water to move through, and some of it's going to stay stored there in the soil. So life in the soil pictures. Is that fun?
Yeah.
Movies are, because, you know, especially when we're going to be looking through the microscope on Friday, that movement, it makes it so much easier for you to find these organisms. So, what organisms are in my soil? So when we're going to start thinking about sampling your garden, your golf course, your fruit trees, your vegetables, exactly what do we have to be thinking about? We want to first discover what's missing in your soil. Is anything missing in your soil? Do you have to put biology back there? So how are we going to approach this? We'll learn how to, we'll go through sampling soil to determine your missing biology. Well, it's really not that different from sampling soil to determine your chemistry. It's just that soil chemistry looks at soluble inorganic nutrients. For soil biology, we're looking at the organisms. And which one's more important? We're going to be going through that whole discussion. Once we figure out what's missing in your soil, now we want to add those missing organisms back into your soil. And so we got to make good compost. We have to be looking at the compost and figuring out, are the missing organisms in this compost or not? Why use a compost that doesn't contain the organisms that you need? Don't waste your time. So I think it's very important to be looking at the compost and making sure that what's not in your soil is in the compost. So you're putting back in what is missing. ACT, aerated compost tea. AACT, actively aerated compost tea. So which acronym do you want to use? Whatever one you want. I don't care. And it's a little redundant, isn't it, to say aerated compost tea? It's because by definition, if it's composted, it has to be aerobic. But people like to differentiate between anaerobic stuff and aerobic, so adding the A or the AA, whatever, have some fun. Inoculants, you know, can we put inoculants back into our soils? Well, do you know that that one species of bacteria is missing? That one species? How much are you going to improve your soil if you put one species back in? How many species do you need?
75,000.
Yeah. And you're going to put one back in there? Make me laugh. So when we start looking at things like EM, how many of you know that one? So EM stands for effective microorganisms. How many microorganisms in here? How many? Yep. I heard an Elaine. No. 8 to 10. Okay. So typically, you know, a maximum maybe 12. And the species that are in EM. Where does EM come from?
A lab.
A laboratory. And it was Dr. Higa in Japan that developed this particular inoculum. And what he did was look at, what is it, bokashi? That's an H. And he noticed that when people were eating bokashi, and they threw out the liquid that was left in that container, that wiped out a lot of the disease-causing organisms in some of the agricultural flips. So clubroot in cauliflower wiped out when that liquid from the, what is bokashi? Bokashi, basically you take a pot of some kind that you can seal closed. And you're going to take your cabbage and put your cabbage in there and a little bit of salt water. So what's the percentage salt? It's about 15% salt water. Put your cabbage in there, seal it closed, and bury it, because this is going to stink to high heaven. Kimchi, another version of it. Every Southeast Asian country has its version of this, you know, so it gets pretty smelly. And then what Dr. Higa noticed was the liquid, after you eat, how can you eat that cabbage after it pops culture? You have to eat it from childhood. I mean, that's the only way to do it. Because I can, you know, it's like, come on, everybody else is eating it. They're like, no. Can't put that in my mouth. But the liquor from this, the liquid that's left in the pot, put that out on soils that have no decent biology in it. And you can go a long way in getting rid of some of the disease-causing organisms. So what's really going on? Well, A, it really only works when you've wiped out all the other biology in the soil. So putting in some biology is better than having nothing. But have you gone all the way to fully balanced, everything's taken care of, sustainable? Not in the bun. So we're looking in this material. Think about the conditions where we're doing kimchi, or we're doing the boshi, or what's the other one? Sauerkraut, yeah, the European version of this.
Cocoa.
So this is anaerobic conditions, isn't it? It's cabbage in a sealed pot. The organisms start growing so rapidly, they use up any remaining oxygen in that pot. And so what we're dealing with is a culture of anaerobic organisms. They tend to be lactobacillus. And these are very beneficial bacteria from a certain point of view. They're going to drop your pH, so they're going to end up with a pH of somewhere around 2 to 3. Okay, so that liqueur coming out of this material, pH. But they outcompete human pathogens. Even in anaerobic conditions. So if perchance your cabbage had pathogens, the guy putting the cabbage in the pod did not wash his hands after he went to the bathroom the last time, and he's inoculating a whole bunch of you don't want to know in there, the lactobacillus grows and it outcompetes those human pathogens. So this material is going to be pathogen-free. So it's one way of making sure that you don't have the bad guys in there anymore. We typically have a boatload of yeasts. These are the anaerobic forms of fungi. So not the good guy fungi that we need to make things better in our soil. We can have some bacillus species. And these tend to be things that are repugnant to insects. So you have these bacillus, you throw them out in your soil, and all your root-feeding grubs and your root-feeding larval stages of insects, your wireworms, they all head for the hills. Because that bacillus is extremely repugnant to those insects, and they're going to leave. So you can have some good impacts, but is it going to bring us all the way to sustainable? Where's the protozoa? Where are the nematodes? Where are your fungi? So this might be step 1 in a 12-step program. You didn't know that this was a meeting of Pesticides Anonymous. So what are the organisms in effect of microorganisms? Well, when you go back to some of the things that Dr. Higa has written, he says, in order for it to actually be EM, you have to have, by definition, EM has to have cyanobacteria in it. The particular kinds are purple, non-sulfur, photosynthetic. Cyanobacteria, that's its name. How'd you like to have that thing for a name? So, purple. photosynthetic. So the photosynthetic pigment in this organism is purple in color. It's not your typical chlorophyll. It does require sunlight in order to grow. And so when you get a culture of EM, if you listen to Dr. Higa, it has to have this cyanobacteria in it. It's got to have a bunch of different species of lactobacillus. It's got to have some yeasts. It better have some bacillus species, and oh yeah, I forgot one. Actinobacteria. If you don't have all of this diversity, it's not EM. So time and time again, at Soil Food Web, we have ordered EM from places selling it. And the first thing that we do at Soil Food Web is we open up the jug and we put a drop on our microscope slide and we take a look. 50% of the time, it's water. There is no life at all in that material. So do you see why EM gets a bad name? But when you put water out, what does it do? It does not kill anything, does it? It doesn't help in any way if it's just sterile water. So we invariably then call up the company and we said, we ordered some EM from you. And now EM is supposed to have blah, 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 blah. Here we go. And when we took a drop, and we actually typically take 10 or 20 drops looking for something, anything, and we couldn't find any life in that material at all, we would like you to send us a sample of EM, not water. This is really expensive water. And I always love them, because their first response is, well, you couldn't possibly tell them. Ooh, do you know who you're talking to? Well, you can't tell if those organisms are in there or not. Oh, yes, we can. Let me send you pictures. And then they, so we back and forth, and then almost invariably, they'll send us another jug, and we look at it, and there's no life there. And then they send us another jug again. Finally, we'll get a few lactobacillus, but you really do want to be looking at this stuff when you order it, so you know that you're actually getting what you paid for.
Why is their kimchi not producing?
Because there is no quality control in the company. They are not looking at this stuff at all. So they're just going through the motions. How many times have you put a recipe for bread together and you put it in the oven and it just went, yeah, but I did everything right. I did everything the recipe told me to do. Yeah, well, it's the results that count, isn't it? So we've got to see the organisms. You got to see that bread rise. You have to see. So look out for inoculants. Please be looking at those through the microscope and make sure the organisms are there and that you're not being passed off something that's not really life. What you're paying for is life. There are a number of different biocontrol agents, and we will go through those different biocontrol agents on Thursday when we talk about compost and compost tea, because that's the easiest way to get biocontrol agents to actually work. Most of the time, the biocontrol agent comes in a jug of some kind, a container, typically dry. So you've got spores or you've got cysts of the biocontrol agent. So Bacillus thuringiensis, bacterium, that makes a protein that is toxic to caterpillars and other insects. And so when you buy a jug of BT, you're buying the spores. If you just take a handful of that dried material and throw it out in your field, those spores are not ever going to germinate. They're not going to grow. You're not going to get any benefit from having wasted your money that way. You really want to do something that will wake those BT spores up. And the best way to do that is to put them into a compost tea and get them to grow in the compost tea. One of the food resources for Bacillus thuringiensis is humic acid. When you have a bunch of trichoderma, a fungus that attacks other fungi, you have an outbreak of Phytophthora, you have an outbreak of mildew, go get some of those spores of trichoderma, throw them in your compost tea because humic acid wakes up those spores of trichoderma. Now spray your leaves out and in 12 hours, the mildew's gone. you gotta wake up the trichoderma first. Trichoderma, you buy it in most garden stores. Trichoderma. Hey. And we will go over these again in on the compost day. So we will. Yeah, there will be a handout on this. So trichoderma, this is a fungus. It is parasitic on other fungi.
So you add it to the compost tea.
Yep, we will put that in with your tea. Because during the brewing process, these spores will germinate and grow, and any fungus that it comes in contact with, gone. Think about that long too. Okay, 20 minutes. Even beneficial fungi. Now look out. What if you put mycorrhizal spores into your soil, and you're trying to get mycorrhizal colonization on your root systems? And then you put in a bunch of trichoderma activated to wipe out all your fungi. They don't care. So yeah, maybe use the trichoderma first to take care of any diseases that you've got, and then come along with your compost tea, and put that really good fungi back into the system. So trichoderma, when it's out there in the real world, typically 2 weeks after you've added it, wiped out all your fungi, they go to sleep. They won't wake up again because they're not in there with a high concentration of humic acids. Not until the trichoderma run across those particular conditions. So I'll go through a number of these biocontrol agents. I can't keep up with them all, but it should serve to make you be able to think about what you need to think about. Dipel, what is that? That's what is dipel? Okay, so it is BT, Bacillus thuringiensis. That like serenade is another bacillus species of Bacillus. Something or other.
Is whey essentially the same thing?
No, whey is just the protein liquid when you're making cheese.
They have actually, so it's
It might. What cheese production facility is that coming from? Which? Now, what cheese are they making? And so, if that's a cheese that's used by growing a whole bunch of fungi, is that whey going to have that bacterium in it? Yeah.
Not familiar with the process.
So you might need to know a little bit more.
Because that's just a source of lactobacillus and natural farming movements, and so I'm curious if I could do the same thing.
So what's the easiest way to tell if your whey actually has the lactobacillus you want in it?
What was the answer?
Microscope.
Oh,
Yeah. Must have microscope. So, we want to have those people that can help you use these microscopes. Oh, wait a minute, I didn't get done with the last one. So now you add the organisms, you add a whole diversity, bacteria, fungi, protozoa, nematodes, microarthropods, everybody. Same thing in an actively aerated compost tea. Inoculants, maybe a few species, biocontrol 1 species typically. Add them into your soil. You might put them in with foods to help them grow a little bit. So what foods? We'll go over that on Thursday. And then monitor to make sure that whatever you've added, it's actually growing. We deal with a lot of soils where there's some nasty toxic material left in those soils. Number of places where people have put out dildrum, or they've used Saigon, or they've used, you name your favorite pesticide. And that's accumulated over the years. You can have accumulations that are so high that it'll kill your first inoculant of microorganisms that go into that soil. How do you know that happened? You gotta use your microscope. So we didn't have the biology, we added the biology, and 2 days later it's all dead. Maybe you'd like to know that, because then we're going to have to do some different things. We're going to have to go in with some good food resources to help those microorganisms overcome that toxic load of pesticide, inorganic fertilizer, salts. Keep adding them until we do get rid of those toxic materials. any pesticide you want to talk about can be decomposed. There is no pesticide on this planet that we haven't found an organism that will decompose it. Sometimes it takes us a little while to do that. So let's make sure we get all of that organisms back into the soil. So typically, I like to start it at the end. Start doing this at harvest. You go and harvest your crops. And so once those residues go down, we want to apply the organisms to the soil, to the residues, so that those residues get decomposed as rapidly as possible. Fruit trees. When do your fruit trees drop their leaves? Now, mango is like weird times of year that mango drops its leaves. How about papaya? Or how about coconut? Well, year round. So anytime you want to with some of the fruit trees, you go out and apply this biology to the leaf material that's fallen into the soil. So we start improving that soil structure. Certainly before the rainy season, you would really want to get good structure built back into your soil. A lot of times we're dealing with early successional weedy species growing around your trees and really damaging the ability of your plant to take up nutrients. So let's get out there with some cardboard. Cover those weedy species with cardboard, put your compost on top of the cardboard, and then go out and let's establish some really healthy understory plant species that have the same mycorrhizal fungi, have the same ratio of fungi and bacteria that your fruit trees require. So we're going to set the stage to have really productive fruit trees once this all gets established and growing. But a lot of times this grass can be so pervasive that you're going to have to do something to knock it back. So something, not a toxic chemical, put it down over your existing early successional plant species under your trees. So you suppress them, but then you've got to put in a plant that's going to prevent those grasses from coming back. Get some competition in there. Make sure they're short, low growing, good nutrient cycling, exactly the ratio of fungal bacteria that your above-story, above-ground plant requires. Same thing when we're dealing with your gardens, when you're dealing with agricultural systems, especially in this part of the world where you have massive rainfall events. We cannot have bare soil because you're going to lose all your soluble nutrients if you do. So what are the low-growing understory plant species that are native, natural in this part of the world? Most of our vegetables, groundnut is often a possibility. You just have to be careful that you get a groundnut that doesn't vine up your plant and pull your plant over. It's a little depressing when all your onions all of a sudden go because the groundnut decided to grow up it and over it goes. So then we've got a little bit of work to try to figure out what some of those understory plant species might be in this part of the world. But we've got it pretty much figured out for the rest of the United States. Okay, how about if you all go home tonight and look at your favorite native plant website, and you look for those plant species that only grow 1 to 3 inches tall. They don't vine, so they're not going to go up your plant. No kudzu, sorry, no, don't allow that. Probably no vetch, because vetch vines. Clover. But we want to mix, because anytime we are dependent on one plant species, there are going to be those years where it's not going to grow. The weather's not right for it, so we want a mix of these underground, below ground, understory, low-growing, mycorrhizal. Let's make sure that they are mycorrhizal the same as what our vegetables require. So we never have bare soil. You will never till your soil. You will never till your garden completely, ever again. No bare soil. When you harvest in the fall, you'll let those residues fall on the soil surface because you've got really good plant growth growing in your driving rows and your walking rows. Those residues are going to decompose very, very rapidly. These understory plants grow back into the rows. So now you have complete ground cover. You will never have leaching, you will never have loss of your nutrients, you will never have weeds coming back into that area, because we got to choose those things that will not allow weeds to invade into that area. You won't ever have to mow again. How many of you would like to stop mowing your weeds or tilling your weeds? So let's get rid of all of that work.
So what are the not similar to like for trees or vegetables, like rhinopina, what is this?
So we're going to have to, yeah, I'm going to have to get all of those species from you that you might want to suggest. I may not know the answer. We may have a little bit of work, so some of you might have to bring some soil samples in on Friday so we could look at what the balance of fungi, bacteria, protozoa, and nematodes are.
You got mycorrhizal relationships.
Yep. Are they mycorrhizal or are they not? Well, a lot of times you can think, well, there will be an answer, a simple answer that I will present to you probably sometime tomorrow that will help you figure out this answer. So another cliffhanger. Stay tuned, you got it back tomorrow.
In my experience, just the grasses have been so aggressive that anytime I try to like establish something like this, the grasses just come back and take it back over. Is it just that my biology isn't getting up to speed for that, or?
Yep, there's something wrong with biology. Your soil is selecting for the grass growing instead of your understory plant.
Yeah.
So let's look at that.
Okay.
Yeah, so
Like this guinea grass, it likes most everything. I don't know, maybe it does.
that tall grass out there,
Yeah.
very bacterial. As soon as you start getting fungi back in that soil, that grass will come back.
Really?
Really?
We see that with Bermuda grasses. Does not come back. So we gotta get the fungi out there.
I noticed that because my garden is a clearing in the forest, I don't have any green grasses or pumpkin grasses or anything, because it's mostly fungal algae.
You didn't destroy that fungal dominance in your soil when you established your garden.
Yeah, we didn't tell about that.
Yeah, you didn't till. You go, what does tillage do to the life in your soil? So imagine Paul Bunyan coming through here with a great big plow. What would happen to this room if a great big gigundo plow came through that back wall? How many of you would survive that experience? Not many of us. And those that did survive that experience, how soon are you going to come back and take another class in this building? Even if we rebuilt it. So when we do that to the organisms in your soil, when you till, think of the destruction. We're destroying all that structure. There is no way for oxygen, for water to get back through that soil. We just sliced and diced most of the fungi. We just crushed all the good guy nematodes. We just wiped out the good guy protozoa. What's left? Bacteria. You have a strongly bacterial dominated soil. Who are we setting the stage to grow? What plants require strong bacterial dominance? Weeds. So think about this one. If you did nothing to your property, if you did absolutely nothing this year, what plants would grow? And if your answer is weeds, early successional Bermuda grasses, the zoysia, yeah, you've got strongly bacterial dominated soil. And Mother Nature's telling you that. So when you're having a problem getting rid of these early successional plant species, something's severely wrong with your life in that soil. Let's get some fungi in there. Let's get the protozoa back. Let's get the good guy nematodes back in the system. Let's get the microarthropods back in there. Let's get the mycorrhizal fungi back. And how are we going to do that? So we're going to add those organisms. Typically compost. If you don't have enough compost to put out a ton an acre, then we're going to turn that small amount of compost into extract. Or we're going to put it into a tea. What's the difference between extract and tea? So, compost, you gotta have that to begin with. You gotta have good compost with all the right organisms in there. The inoculum has to be present. We take that compost and put it into a bag with nice holes in the bag. You can take that bag put it into some water and massage it really nicely for about 30 seconds. You know, arm power. Massage it. You're giving somebody a really good, strong, deep muscle massage for about 30 seconds. And we're going to get most of the organisms in that compost into the water. We've extracted. So that's a compost extract. Question, do we have enough life? So is that enough? So take a drop of this, look at it in your microscope, and if we're not seeing, in our field of view, we want to see one fungal strand in every field. We want to see a couple thousand bacteria, nice aggregates, in every field. And in every field, we'd like to see one flagellate or one amoeba. Somewhere in that whole drop of water, we'd like to see a couple nematodes. So, do we have enough? If we just extracted, say, 1 pound of compost in 5 gallons of water. Do we have enough? If not, we dump out that compost that's been extracted, put that back in your compost pile. Another pound of your compost goes in your compost bag and massage away, 30 seconds. Pull it out, take another look. Do we have enough biology in there? No? We go and dump that compost out, we put another pound in there. And we just keep doing that until we get the biology up to where we need it to be. And then we put that on our soil. So this is a soil application. Or if you don't have enough good biology in your compost, put it in your compost. Up that biology in the compost, get that going. When we're dealing with a tea, slightly different. Because tea only goes on your foliage. If you're going on your soil, do extract, because it's way easier. You know, this takes what, 5 minutes to make? Tea, we gotta have active organisms, so they stick to your leaf surfaces. So we're gonna protect your leaves, your bark, your fruit, your flowers, all parts of the above ground part of that plant. We have to have active organisms to stick. So we only use tea for that reason. Now we can use the same machine. So there's our compost, and typically 1 pound in 5 gallons. But this time we're going to put an aerator in there. So we're going to have a nice air pump. And typically, like in a 5-gallon brewer, we're going to have a 500-gallon fish tank pump. At least. Maybe more. And all we're going to do is run an air tube, just a nice tie-on tube down to the bottom, and use a suction cup or something to hold that on the bottom of your bucket and let her go. Turn that aeration on and let the surface of that water just be rolling boil, just like you got the stove top going full strength to get that water boiling to make your tea. Just as fast as you can. So we're going to be extracting these organisms out of that compost into your water. It takes a fair effort to pull those organisms off the surface of your compost. So if we're not going to do this hand strength, we're going to turn on the brewer and let this brew. And then we're also going to add foods. Thursday we will go over the foods. How much and what kind of organisms and all that good stuff, so stay tuned for that.
How long do you boil it?
Typically, we're going to add the foods. So let me go through this step by step. We're going to add the foods. Make sure that this is all nice and rolling boil. And on about hour 6 to 8, we're going to take the bag out. We don't have to keep extracting. And once that, when we're doing this mechanically, we can take this out after about 6 to 8 hours. But then we're going to let the organisms grow for a full 24 hours. When it's warm, when it's nice, warm day out, typically 24 hours. What if it's a little bit later in the season in Minnesota? We're going to let it brew for 48 hours. What if we're brewing on Thanksgiving sleep in Pennsylvania. We may well be letting this brew for 72 hours because it's the growth of the organisms that's important. We want to see one strand of fungal hyphae. We want to see 1,000 bacteria. We want to have at least one protozoan. So see the similarity here between what we want in an extract as what we want in a tea? Same numbers, so that we can cover those leaf surfaces. I'm not sure how many plants are still leafed out in Pennsylvania in November, but if somehow. So you brew at the temperature of the plants that you're going to put things on. I want to protect the surfaces of my poinsettias, and I'm going to put them outside. So I better be brewing at outside temperatures. Okay, my poinsettias are going to be inside in my house, so do I brew at outside temperatures? No, because if I brew outside in the middle of November in a cold climate, I'm going to have bacteria and fungi that like to grow where it's cold. You bring them inside, and those cold-loving bacteria and fungi, you spray them out on the surface of your poinsettia that's living at 72 degrees. What do those organisms do? It's too hot. I'm just going to go to sleep. I don't like it this warm. And now you're going to have to wait while your warm-loving bacteria and fungi wake up and start doing their thing. The disease is wiped out your poinsettia. So you brew at the temperature of the plant you're applying this thing to. I know there are tea brewer people out there that say bacteria and fungi grow faster at high temperature. So if you want to have lots and lots of bacteria and fungi, you should be brewing at 85 degrees. It's not going to do you any good if your plant is growing at 75 degrees. Those organisms are going to go to sleep. Do you no good. So you see why compost tea fails sometimes? People don't listen. You don't brew at the hottest temperature you possibly can. You brew at the temperature your plant is. I love it when the guys in Las Vegas, they get these tea brewers. And they put the tea brewers outside because they're going to be spraying their palm trees and their cycads and their lawn and everything else. And they're out there starting the tea brew, and it's too hot. They don't like having to be outside when it's that hot. So then what did they do? They brought up their tea brewers inside, and they were brewing at 72 degrees. Yeah, organisms that grow at 72 degrees how long are they going to be alive and active when we blow them out there at 115? Not. So this tea stuff doesn't work. Yeah, this is bullshit. This doesn't work at all. Right. You know, you lose some, you win some. So, really getting the organisms out there. I don't care whether you use compost, whether you use extract, whether you use tea. The point is to get the organisms out in the system. So, here we go. We put them in when we harvested. Let's monitor and make sure that we're going in the right direction all through the non-growing season. Now, come the next spring or the next time we're going to start planting here, we want to look at that soil and, gosh, if we got to go out there with more compost or extract or tea, okay, pre-plant, go out and get that on. Apply the organisms to the seed. So when we're seeding something in, or what if you're putting in bare root trees? Make a good tea. Soak your root system in that compost tea so all these organisms are gluing themselves onto your root system. Make sure you put the mycorrhizal fungi in there if your plant needs mycorrhizal fungi. Get all that on the root system so when the organisms germinate and grow, they're right there in the root zone. Castle wall, protecting things. Seeds, same thing. Let's get the organisms all on those seeds. So you soak your seeds for about 15 minutes in that compost tea. But now take that out, dry them down. If your seed box, if your planter can't deal with wet seed, then dry them down. These organisms are going to be in all the cracks and the crevices in the seed coat. They're protected from being dry. They're going to hang out. They're going to be just fine for a couple months. So you plant with the organisms that you need going into your soil. And then, be checking your soil, making sure that, you're getting that biology going. If you have any problems above ground, and think about the fact while we're still trying to fix things in the soil, you may have to put on a foliar application to protect from the diseases that may still be in your soil, the pests that may still be in your soil. When people have looked at fruit trees, 70% of the diseases and insect pests that attack your fruit trees come from your own soil. Those eggs were laid from the adults last year, earlier in the spring, and they haven't gotten wiped out by doing what we're trying to do, fix the biology in the soil. You're not there yet. So this last year, last time you may ever have to put a foliar application of biology on your leaf surfaces to protect your plant. But in this transition, while we're trying to get things right in the soil, we need to be putting that biology on the leaf surfaces to protect you above ground. What if we put the biology out and after you plant your seeds, or after you plant your bare root plant, trees, shrubs, whatever, and you're looking and seeing that we don't have biology where we need it to be yet. We're moving in the right direction, but we're not there yet. Okay. Extract. We can be putting extracts out as a liquid application. Put it out in the irrigation. Put it out when you water. Pretty easy to go out and spray these things. Instead of spraying pesticide, let's be spraying the biology. So that kind of gives you the basic outline of how we're going to approach this. When we start out working with somebody, we have a slew of questions we're going to want to ask you. Where are we starting from? So you've got a list here. What do you want to grow? So what is the proper balance of fungi, bacteria, protozoa, and nematodes? What problems have you had in the past? So what diseases, what insect pests, what problems are you dealing with? Poor fertility. The root systems are only going down 2 inches. We're going to check for compaction problems. How do you check for compaction problems? Penetrometer. Do you all know what a penetrometer is? Basically what a penetrometer is a metal rod. David, do you have a penetrometer? Okay. So take a metal rod. Got that? What is that? It's about half an inch. Half-inch diameter metal rod. Stainless steel. When you walk out into your field, this is real tricky, you push it into the soil. And as you start having problems to push that through the soil, guess where your compaction is? Right there where you can't push. So, what's the maximum weight I can actually push through the soil? About 300 psi. When I hit about 150 psi, most plants cannot push their root systems through a soil that is compacted that much. So I start pushing, and as soon as I have any trouble at all, as soon as I start going, my root system, that's probably as far down as it possibly can go. Now by the time I get to 300 psi, there is no plant that can push its root system through something that's 300 psi. Even like a dandelion, something that's got a taproot that can push its way through some really nasty stuff, generally can't make it through 300 psi. Some really good work from University of Pennsylvania showing these relationships between where most plants are going to get stopped, where most taproot plants are going to get stopped. So where are these compaction levels in your soil? Well, you don't have any. That would be fantastic. Because then your roots are going to go down as far as they possibly can. So do you have compaction problems?
I've noticed another way to do it is just plant some daikons and then they can go in and just start growing up.
Now what happens when it gets really hot and really dry? You have dead daikons. So do you need to take care of that compaction layer? Get your biology down there. How are we going to make sure that we get the organisms down to however far down that compaction layer is? We might, one last time, you might have to till. So you spray your organisms on your soil surface, or you put your compost out, and you come along, and the last time you will ever till your soil, hopefully. So you start this whole process of going sustainable. And then plant your understory plants in there, put in your above ground cash crops, strip till into your understory plants.
I've heard of people injecting.
Yep, that will work as well. Now, typically people are going to inject where you have a plant, a perennial plant already planted in the system.
Such as an orchard.
So it's just an orchard, right? Or blueberries or raspberries or something like that, where you really don't want to have to go out and rip all your plants out and then replant. Not fun. So that's where we might inject. So where is that compaction layer? Where do you have to get that biology down to? A lot of times golf course, turf. We don't want to pull all of that up and replant. So we're going to go in and do an aeration. So when I talk about aeration pores, have you all seen that on golf courses where they go with big machines that have like 30 little cores, and every other one punches into the ground, and then the next set punches. Well, these guys come up, spit all those cores out. So punch, 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 all the way across the golf course. We're aerating. We're opening up that compacted sod, that compacted layer of turf, to get oxygen to go into the soil. Quite often then they'll come along and fill those holes with a mixture of 30% compost and 70% sand. We've even gotten the U.S. Golf Association to suggest to their golf superintendents 30% compost, 70% sand. 15 years ago, if you said the word organic matter to a U.S. Golf Association superintendent, you would go absolutely ballistic. It'll kill the turf. It's just terrible. We've proved time and time again. All that compost stuff just kills your turf. I don't think they were using compost. They were using stinky, smelly, putrid, horrific, awful stuff. Putrefied organic matter. A little bit of a problem there. Of course they're going to kill their plants. So see where you got to define what compost is really clear?
Quick question. What about earthworms? But earthworms are top of the pyramid, so if nothing's healthy underneath, then just throwing earthworms doesn't do much, right?
Yeah, earthworms die really fast with nothing to eat. What do earthworms actually eat?
Organic matter.
Organic acids.
No. What do earthworms actually eat? They eat bacteria, fungi, protozoa, nematodes, microarthropods. Earthworms do not make the enzymes to break down organic matter. They have nothing to chew up cellulose. They have nothing to chew up lignins. They have no way to deal with any plant organic material. They are not decomposers. They're shredders. They take bites and they break that plant material up into smaller and smaller bits. But what they actually eat are the bacteria and fungi, protozoa, nematodes, microarthropods, anything that fits in the mouth and they can crush. That's what it is.
So throwing a bunch of earthworms on a field is not going to help your aeration that much?
It depends. Okay. It depends, really. What kind of earthworms? Which earthworms are the ones that survive here in Hawaii?
Blue ones.
The blue ones? Or the tiger worms? Yeah, so you want to make sure you've got the right ones. Now, tiger worms typically only grow in organic matter. They don't grow in soil. You throw a bunch of the Eosinea out on your soil surface, they're going to be going, what is this junk? And they're heading for the layer of organic matter someplace. And they're going to be tromping along and looking for a nice litter layer to be consuming. Now, Lumbricus, you start dealing with your native true earthworms. Here in Hawaii, where do they live? They're not up here at the surface of your soil. They will come up, grab organic matter from the surface and pull it down their burrow and they line their burrow with that organic material, and they eat the bacteria and fungi that they grow on that organic matter in their burrow. Isn't that fun? So those kinds of earthworms, yeah, they can be aerating your soil pretty nicely for you. But and golf courses, they don't like them, because as they come up to the surface of the soil, they push out in front of them all of the soil from the tunnel that they're building. And so they leave this little ring of soil sitting at the top of their burrow, called a midden. And golf course superintendents hate them, because it's a great way to lose your job. When you're golfers that are paying $150, $200 a round come out here and they putt, and hits one of those little and the ball goes, meow. Yeah, well, so much for that golf course and the superintendent. So, yeah, you have to change the earthworms in that soil. We want horizontal burrowers, not vertical burrowers. Different sets of earthworms. We have a whole half day on earthworms.
Question on when you said no till, what about spaders? Is that the same thing?
They don't damage the soil as much. So now, when it comes to looking at different kinds of tillage equipment, you may have to understand which ones are less destructive, which ones are more destructive. So there's a whole gradient here. So what should your answer be? It depends. When we deal with moldboard clouds, not very destructive. Spaders, not all that destructive. Chisel plow, woo. A disker, ouch. Rototill, you just destroyed everything in your soil except for the bacteria. You've destroyed structure, fungi, protozoa, nematodes, all of those guys are gone. What's going to come back in? Diseases and pests. So you got to look at how much damage to the soil occurred based on the kind of tillage that you perform. You till in the springtime when it's real wet, massive damage. How about if it's real dry at the end of the growing season? Big deal. They weren't active anyway. So lots of these kinds of factors that you have to put into it.
What was wrong with the chisel plow?
You're slicing and dicing. Yeah. So chisel, you're getting we've seen lots of destruction. Plowing and coming through. The chisel plow is way scarier than a moldboard plow. So how much damage? How do you know which one's more damaging and which one's less damaging? Microscope. Take a look. You can find out for yourself what happened to the biology when you did any kind of tillage.
So if you have compacted set of soil, say just like your kitchen garden, it's a small space, you don't necessarily want to bring in, you don't want to till at all. Would you suggest just working up? Just like maybe adding logs and rocks and just kind of filling up from there, because you don't want to get in there.
Yeah, or for the last time ever, you're going to do a double dig. So Jevon style. One last time. But get down there.
How deep do you need?
You probably want to go to where you're below any compaction level that's up in these ranges. So you want to dig down that far. And so if you're digging by hand, make sure then that you add the compost or your extract or your tea in there. So throughout that whole profile, down below your compaction layer, you're getting that good biology in there. And then you're just going to go down the line, make sure that biology gets in. Let's plant your understory plant species, or at the very least, put in some mulch.
But what if you already have established perennials and things, and that's just not an option?
Right, that's where injection comes in.
Injection.
Yeah.
So
you
that's
already
just
have
like
perennials.
a giant syringe down into the
And there's all kinds of different injectors. And in my experience, they all work just as well. I just really haven't noticed much difference. So it's really what size, how much. For those of us who are real cheap, I just get a nice soil core, or I'll just go in there with a bulb planter, pull out that, the top layer, pull it out with my bulb planter, and then get the next one and get the next one, and just make sure I spray lots of really good compost seed down there where they're beyond the compaction zone that I can measure. And get that all covered with that real good set of organisms from my tea or my extract. And then a mixture of 70% sand or or 70% site soil mixed with a good compost and fill the hole with that. And you're getting the organisms down there, and of course organisms. Are organisms only going to go one way? Every direction they possibly can, and down, and up, and they're going to move out from those injection sites. So just be testing your soil. How fast do your organisms move from a core that you filled with that really good biology? Very good. It depends. Did you really have good structure in your soil to begin with, or was it totally compacted? How much rain? How many roots do you have down there feeding the microorganisms? So lots of factors.
So just a few feet apart, just use it.
There are experiments that we have done looking at one point in the middle of a field. And from that one point, we could show that the biology spread half a mile in every direction through that one growing season. And I can also show you places where we put the biology into that soil and it didn't move at all. What's the toxic chemicals in your soil? So if you haven't been working with toxic chemicals and you know that nobody's used a toxic chemical in your soil for years, it's probably going to move really fast for you. If you don't know what they did to your soil previously, you're going to find out what's in there. So we typically want a map of your soil. If we're going to work with you, you start working with somebody else, they generally want to have a map of your soil. You know, where do you sample from? We want you to focus on the plant that you care about. What is the plant you want to know something about? Is it your broccoli? Is it your mango trees? Is it your bananas? Is it your, what is it? And now, we want you to go to your field and pull soil pores from at least 3 areas and consolidate those pores. You always want to be picking at least 3 places to sample from. At least 3. You don't go to the middle of your field and sample right in the middle of your field and say, I've got a representative sample from this field. Because what if right here in the middle of your field is where the mouse pooed last night? What are you measuring? Mouse poop. What if that's where they buried the mule 20 years ago? What if this is where the cat dug last week and left you little gifts? So you want to be sampling from a number of places. So choose an area where the plants are growing and more or less all very similar, and pull samples from at least 3 areas, 3 points within there. Wouldn't 5 samples be better? You would be more representative. Wouldn't 10 samples be better? Wouldn't 150 samples be really? Okay, wait a minute. We want to have some soil left in your field. So, your choice, but at least 3. So we get some idea of representativeness. Now, when you choose from all those possible places, you go and take your samples. But you need to focus on the plant that you want to know about. So we had our field and we are going to take a sample from right there. We're going to take a sample from here. And we're going to take a sample from there. Random, right? So once we're out in the field right there, looking at the map of our property. Once we're out in the field right here, I want to find the closest plant I care about. And I'm going to walk over to that plant. And now I'm going to go halfway between the drip line of that plant. So here's my tree. Where's the drip line on my tree? So where a drop of water coming from the sky does not contact your canopy, you're outside the drip line. Where a drop of water falling from the sky contacts the canopy of your plant, you're within the drip line. So here's my drip line. Over here, you know, it's not going to be uniform. It's not going to be a nice, perfect circle. So define what that drip line is. And now I want to go halfway between the stem of my plant and the drip line. So there's halfway. And so right there is where I'm going to be taking my sample. Doesn't matter on the size of the plant, because we already know that this is going to be way more fungal right next to the plant. And the further out you go from the stem, you're going to have fewer and fewer fungi. So how do we make it uniform, no matter what size this tree is? How do we know where we're going to have that same ratio of fungi to bacteria? We always go halfway between the stem and the drip line. We're looking at that same ratio, so we're not getting a sample from over here where it's bare soil, and a sample where we've got a bunch of dandelions. We're going to be pretty uniform if we go halfway between the drip line and the stem. Okay, here's a good question for you. I've grown some chard. Where's the drip line on my chard? Right, because, you know, there's my drip line on my chard, there's the stem, so halfway in between, it's going to be right here. I just want to make sure I'm getting root system, and I'm being pretty standard about the part of the root system that I'm pulling from. Carrot. Where's the drip line on a carrot? Right next to it. So we want to stratify our sampling for the plant we care about. So we take our soil core. I always go and get, this really expensive, really wonderful, high-tech piece of equipment. It's called an apple core. Wonderful thing for taking soil samples. So go get an apple core from the grocery store. Whopping big, what, $2? And so this apple core, the teeth usually go to 5 centimeters, 3 inches. Push it down into the soil halfway between the drip line and the stem. Turn it round, cut those roots as you're turning, lift that out and put that right into your plastic bag. And then go to the next area that you're going to sample from, find your representative tree or bush or corn or veggie or tomato. Halfway between the drip line, stem, and go with your apple core, turn it around, pull that up into your plastic bag. So at least 3 cores like that. So now as you're walking away, break up the core, get all of those nice aggregates broken up as much as you can. And then take that back to your microscope and take a look at it, or send it into a lab and get it looked at.
Is 3 inches your ideal?
Yeah, most of the work that we've done is 0 to 3 inch depth of the soil. But if you got a thatch layer on top of your golf course, push it to one side, we want to start at the surface of the soil. So if you got a litter layer on your soil, push that aside, start at the surface of the soil. How do you know it's soil? It's got sand, silt, and clay, or clay. It's got to have those mineral particles in it. So if it's just organic matter, push that aside, start at the surface of your soil, go down about 3 inches. Well, doesn't it matter what's going on deeper and deeper and deeper? We've already done that. You don't have to repeat it. We already know that biology is going to stay pretty much the same all the way down to the bottom of the root system of your plant. It's once we drop out of the root system of your plant, that's where biology starts to drop off. So if you want to, you can go 0 to 6 inches or 0 to 12 inches, but that's hard work. I don't know about you, but I'm lazy. I don't want to work any harder, and I absolutely have to, which I think is why I'm in this business, because I am the world's laziest plant grower. Break time! So let's take a break and have some fun with this now.
How many minutes?